Hello, in this video I wanna teach Comparative Genomic Hybridization Technique, CGH. Comparative Genomic Hybridization Technique is a very useful technique to detect deletion and duplications in solid tumors. Classical cytogenetic methods, for example karyotyping, are very useful to detect, for example, deletion, duplication, insertion, or inversions in liquid tumors, for example, hematopoietic or mesenchymal tumors. But for solid tumors, this technique is not a perfect one because tumors that are originated from solid tissues are not able to reach a good ratio of division in cell culture. They have a low mitotic rate, so we cannot able to create good metaphase spreads of them. Sometimes their culture could be contaminated with microbial agents too. So because of this inability of making good metaphase spreads, CGH is a very useful technique to detection, deletion, and duplication in this kind of tumors. In CGH array and in CGH technique, we could detect any kind of unbalanced alteration of chromosomes. Unbalanced alterations are the alterations that in them chromosome gain or lose some genetic materials. For example, deletion and duplication are kinds of unbalanced rearrangement. In CGH, we have three kinds of DNA material. We have a metaphase spread of a normal male or female. It is preferred to having a female spread because of having two X chromosomes. This spread is originated from a normal female. Because of the impossibility of having good spread of solid tissues, in CGH we have extracted DNA from tumor cells. So we don't have to culture the cells of the solid tissue. DNA from tumor tissue are labeled by a green fluorescence dye. We have another population of DNA from a healthy tissue. Then we extract DNA from a tumor tissue as a test DNA and a healthy tissue as a reference DNA. We should label both of them. In CGH, labeling of DNA is done by nick translation. In nick translation, test DNA is labeled by biotin, and the reference DNA is labeled by digoxygenin. After labeling the DNA from two tissues, we add cut1 DNA to both DNA populations. Cut1 DNA is a placental DNA enriched with repetitive DNA, ranging from 50 to 100 base pairs. The role of this blocking DNA is to block the repeat DNA in chromosomes, especially centromeric and telomeric repeats. After adding cut1 DNA to both DNA samples, we add streptavidin and FITC or FITC to the test DNA and digoxygenin antibody labeled with FITC to reference DNA. If I wanna to use the exact dye of the fluorescence, I should use the red one for reference DNA, digoxygenin antibody labeled by FITC, that is red, and streptavidin labeled by FITC, that is green, to test DNA. So we have two populations of DNA with two different dyes. After this step, we denature the DNA on the slide and two samples of our DNA separately. So we have a metaphase spread that is denatured and two samples of DNA that both of them are single-stranded or denatured. In this step, we add both of the DNA samples to the spreads. When we add the samples to the spread, fragments of DNA in both samples compete to attach to the spreads or single-stranded chromosomes that are fixed on a slide. So, in many regions of the spreads or of the chromosomes, we have the green and the red signal in equal amount. But in some regions, we may have the ratio is not 1:1. Sometimes we might see an elevated ratio of green to red dye. For example, in this region, we have an elevated green to red ratio that is showing a kind of duplication of DNA in this region in tumor cells. So in tumor cells, or in this sample that is originated from tumors, we have duplication in this region. So in tumor sample, we have more representative of this region than the representative that are in the reference sample, because reference sample is from a healthy tissue. It could not be suffering from a deletion. So we have an duplication in this region just in test DNA. And in some regions, for example here, we have a reduced green to red ratio that is a result of deletion in this region in tumor cells, because there are fewer representative of this region in test DNA than the representative of this region from reference DNA. As I mentioned before, the red signal is originated from a reference DNA, from a healthy tissue. So it could not be suffering from a kind of duplication, for example. So in this region, the reduced green to red ratio is a direct result of deletion of DNA in the test sample. So by this method, without preparing any kind of metaphase spread from tumor tissue, we can detect any kind of deletion and duplication by 20 megabytes resolution. That is not a good resolution, but it's very useful in many cases, especially before inviting the array CGH or CGH array, that in the same manner could increase the resolution, for example, from 20 megabytes to maybe 5 or 10 kilobases, according to the probe size that are designed on the array. Array CGH is a very promising technique that I am teaching in another video next time.